So the title is Long Non-Coding RNA Dysregulation Alters the Activity of Cancer Genes. And what this is going towards is essentially an attempt to systematically test how many long non-coding RNAs have an effect or potentially have pathophysiological effects in cancer. This is a key question in the field, but it hasn't really been systematically addressed. Maybe the most highest profile attempts to address this question appear like in this paper from Riken a few months ago, where their main interest was to measure the expression of RNAs. But buried in one of their figures was a study of GWAS data that suggests that alterations at the loci of hundreds of long non-coding RNAs are predictive of disease state. Our approach was similar to guilt by association, where essentially we looked for long non-coding RNAs that modify the activity or alter the expression of oncogenes, tumor suppressors, cancer pathways, with the idea that if you're able to dysregulate these genes and these pathways, then you will have function that mimics the genes that you regulate. And we developed kind of a big system that looked at long non-coding RNA, predicted long non-coding RNA targets in various models, including cofactor, guide, switch, and decoy. I'll just give a very brief idea of how this works. So in the terms of cofactors, we were looking for long non-coding RNAs to bind and either attract transcription factors, either inhibiting or activating transcription to proximal promoters. In the case of guide long non-coding RNAs, we looked for long non-coding RNAs that bind proximal promoters and recruit transcription factors. Switch long non-coding RNAs are ones that alter the activity of transcription factors, RNA binding proteins, and microRNAs on a set of their targets. And decoys inhibit the activity of transcription factors, RNA binding proteins, and microRNAs by binding them and essentially inhibiting their ability to target their targets. Now, we made these predictions. We first, of course, predicted all kinds of regulatory networks, transcriptional, post-transcriptional. We integrated data from sequence analysis, all kinds of assays like eClip and ChIP-seq from wherever we could find. But I'm not going to get into it in detail, but really the workhorse behind our system is the idea that long non-coding RNAs modulate the activity of regulators. We call these regulators effectors in general. They're RNA binding proteins, microRNAs, transcription factors. And essentially what we were looking for is after we build these types of interactions, we looked to see that the expression of a long non-coding RNA, say we just compare low expression and high expression within a data set. So we looked at tumor data sets. So we'll have hundreds of primary tumors that were profiled for RNA. We look to see that there is a change in the correlation. We use distance correlation between the regulator and the target across multiple regulators for the same targets or across multiple targets for the same regulator. And having integrated this with other data, we would then make a prediction that, say, IRF4 targets PIK3R1, and that a specific long non-coding RNA, in this case OIP5AS1, alters this regulatory interaction. So we build these types of regulatory networks for 14 tumor types, and then we did a simple test. Using ridge regression, we first tried to predict the expression of a target, of every target gene, every gene that's profiled, using its copy number variation and all of the predicted regulators that we have for this gene. We used ridge regression. Then we did the same thing, including long non-coding RNA expression or copy number variation, Then we did a test to essentially establish whether the expression and the copy number variations at low non-coding RNA loci are predictive of target expression after accounting for the expression of all of its predicted regulators and its copy number. And we used multiple methods required, but all of them have FDR less than 0.01, F-test, non-parametric methods. And after all this was done, for every one of the tumor types that we studied, we identified hundreds of genes where low non-coding RNA expression and copy number, so copy number here is in green, expression in red, are predictive. The numbers correspond to cancer genes, so these are tumor suppressors and oncogenes that were combined from multiple databases, but that's the business that they do. And the point here is that for every one of the tumor types we looked, we found lots of cancer genes that we have evidence for low non-coding RNA regulation after accounting for all this other data. Looking at the genes that were predictive in terms of both expression and copy number, the variance that is explained, that is accounted for by low non-coding RNA expression and copy number is not small. It's roughly a third. So we have in the bottom what's accounted for by copy numbers, in gray by all canonical regulators, and the top part is by links. We started investigating these things a little bit more in detail. We found that low non-coding RNAs that we believe are binding proximal promoters have an enrichment at the core promoter. So the binding sites are enriched at the core promoter. So in blue here, you see transcription factors. We know that transcription factors are enriched for binding core promoters. So the core promoter is about 100 bases long, say 50, minus 50 to plus 50 around the TSS. If we look only for sequence signals, this is what we get. If we look at our method for predicting low non-coding RNAs, neither one, both are agnostic to location in the proximal promoter. We see a stronger enrichment. It's significantly stronger. Not as strong as transcription factor binding sites, but significant. Oh, and in fact, the number of binding sites that a low non-coding RNA had in the proximal promoter was predictive of the fold change in low non-coding RNA targets. So since we have transcriptional and post-transcriptional predictions, we started looking to see which ones are stronger, and we found that the vast majority of predictions in yellow here were transcriptional, and these tended to be inhibitory effects. So most low non-coding RNAs that we predicted to target genes target transcriptionally and inhibited the transcription of these genes. So we had this idea, well, what if we look at each one of these low non-coding RNAs and ask whether they're enriched for transcriptional or post-transcriptional regulation? And when we did that, we found a few that did both, more that did either one, so this would be post-transcription, this is transcription, and we compare that to very few, but enough data that suggests the localization of low non-coding RNAs. And we found that enrichment in terms of our predicted transcriptional low non-coding RNAs were enriched in low non-coding RNAs that have been described to be nuclear. Our post-transcriptional interactions were enriched in those that were found to be, before us, cytoplasmic. We looked at pathway regulation, synergistic pathway regulation, suggesting that low non-coding RNAs are enriched for pathways that are associated with proliferation in tumors. And then we started looking for stuff to validate. So we wanted to do RNA-seq, so we selected some of the low non-coding RNAs that haven't been described in cancer. There are a handful that have been described. And looked for those that have lots of interactions in lots of tumors that regulate important genes. Our top one was this OIP5-AS1, which also is preferentially altered in breast and gynecologic tumors. It seems to be especially important in basal-like breast, Its targets are known oncogenes, and they are they're predicted to be significantly moving in response to OIP5AS1 in 2 large data sets for breast cancer, and it's really mostly in basal. 1 minute. We did RNA-seq, so we have significant enrichment for both transcriptional and post-transcriptional interactions for this gene. Then focusing on the genes, cancer genes of interest, we silenced this long-encoding RNA, multiple breast cancer cell lines showing that the targets are responding, because we think this long-encoding RNA is a pan-cancer regulator, so breast and gynecologic. We did the same in ovarian and endometrial tumors, essentially showing the same thing. Proliferation assays showing that silencing of OIP5AS1 mimics silencing of PTEN. And then to show that this is not just one long-encoding RNA, we started doing these experiments in others. TAG1 is predicted to target this signaling pathway. Silencing TAG1 affects proliferation, affects predicted targets. Wnt1-AS is really high in ovarian. Silencing it affects ovarian oncogenes. You see the proliferation, so it does the opposite. It's an oncogene-like long-encoding RNA. OIP5 is a tumor suppressor-like. Here's another one. I'm out of time. Acknowledgements, and I'll be happy to answer any questions.
Questions?
Yes.
Thank you. Amazing work. So you were looking at long-encoding RNAs that have a target on the RNA or DNA, but how about the long-encoding RNAs that just work as scaffolds of protein complexes? Do they still have a target, or can you still find them through this method?
So I think what you're pointing to is what we refer to as guide long-encoding RNAs, or decoys. So the guide acts as a scaffold to bring a transcription factor or a set of transcription factors to a target. A decoy doesn't bring them anywhere, it takes them away. And so every one of those types of interactions has a whole bunch of rules, that they're, a bunch of hoops that they're required to jump through, but both are accounted for by this Longhorn system. Yes?
Carrie Gaskerlin, UCSD. We should all say your name and where I'm curious, this is beautiful work. Thank you very much. What did you use to try to pinpoint the interaction of the long non-coding RNA and its target, especially the post-transcriptional? I know you have the gene expression, and that's kind of a direct readout, but are you able to get that molecular interaction?
So first of all, I don't necessarily believe any specific interaction in any, so if we have regulatory networks for a whole bunch of regulators, I don't necessarily believe every interaction in any given, if you say, is this interaction real? I predict it, but I don't use it alone. So all of the predictions for long-encoding RNA targeting are statistical. They take into account lots and lots of predicted interactions with the idea that need to be tolerant to false positives. So every long-encoding RNA modulation or target prediction sits on dozens of predicted post-transcriptional and transcriptional interactions, and as long as sort of like some of them are real, the idea that this should work. And we've been verifying, so I showed I think 3, I think we did 7 by now. We're verifying these things in the wet lab. It's true, it's not necessarily deep. We haven't done mutagenesis, but the success rate in RNA-seq and in PCR and in Westerns has been phenomenal. So I think this idea works. Now, to predict specific interactions in every one of these networks, we used, again, every one of the networks sits on top of maybe 6 different types of so for RNA binding proteins, we relied heavily on ECLIP, together with motifs, together with expression, and mutation data that suggests that sort of like when something is mutated, the interaction goes away. So there are like a whole line of things that went into every. I'm happy to talk about it offline, but.
Great, thank you very much.
Should we take more questions? Or? Yes.
So I fully understand, so this network-based approach to predict this interaction, that's really nice and fine. But you showed one slide where you said you have an enrichment of RNA targets on the TSS. That would imply that you directly predict the binding site of the non-coding RNA.
Right.
And I don't know how to do this with current technology in a reliable way.
Right. So let me go back to that slide. I really did not speak much in detail. So the curve, this red guy here is sequence-based. The RNA-DNA interaction prediction by Triplexter, which is from Australia, Bailey's group, I think, from what I remember. And so even that, you can see has a significant peak above, sort of what you would. It's not necessarily very, very strong, right? There are lots of, but we thought that it was good enough to use as weak evidence, okay? So we're integrating a bunch of weak evidence. I agree with you that, it's not a solved problem. I don't think transcription factor binding is solved either, so, yeah.
We're still working on it. Okay, let's thank
Okay.
the speaker
Thank
again.
you.